Today I'm going to explain the cell cycle. Why the cell growth is very important for life. Why the cell growth is important? The cell growth is important for development. For example, if the person want to grow, so they should be having cell growth. If the person want to have a hemostasis, so they need to have cell growth. And for the replacement of the old cell with the new cell, so they should be having a cell cycle. The main steps which occur in the cell cycle are DNA replication and the synthesis and the renewal of the organelles and different substances which are present in the cell. There are four different phases of the cell cycle. The first phase is the G1 phase, S phase, G2 phase, and the M phase. Wait a second, it's out of focus. Now it's focused. The G1 phase is the pre-synthetic phase. The S phase is the synthetic phase, which is like SS synthetic and S. G2 phase is the pre-mitotic phase, and the M phase is the mitotic stage. Okay? There is a stage which is known as the G0 phase. The G0 phase, the cells which are not involving in the cycle, they are inactive, and they are present in the G0 phase. So there in the cell cycle we gonna have the activators as well as the inhibitors. So the cyclin is the protein which initiate the cell cycle, and there is an enzyme which is known as the CDK, which is the cyclin dependent kinase, is the enzyme which initiate the cycle, or which initiate the cycling by phosphorylation. If there is no phosphorylation, they won't be having activity, and if the phosphorylation occurs, so enzyme will stop its activity and detach from the cycling. We're gonna have different type of cycling. As you can see, I have written it. We in the book, we have 15 types, but in the book we only have we have D, E, and B. Okay. There are some checkpoints for the cycle. For example, if the defective protein is forming, or if the defective cell is forming, or there is any abnormalities, so it could be like it should be stopped. So what happened? There are checkpoints. The first checkpoint is G1S, and the second is G2M. The G1S monitors the integrity of the replication, and the G2M, it monitor or ensures the accuracy of the replication. If the replication is not accurate, there won't be any replication, because there is a checkpoint which stops that division, because the replication is not normal, so there won't be any proper development of the cells. Okay, we can have different type of enzymes. Okay, wait a second. If the damage or abnormality is too much, so what will happen? Either the cell will go into the programmed cell which is known as the apoptosis, or either the cell is gonna be converted into senescence, which is the inactive form, or which is the that's it, through p53 dependent mechanism. There are different type of CDK inhibitors. So we gonna have one family which is known as a CDK 1S, CDKIS, 3 proteins, P21, 21, 27, and 57, and the other family of the cycling CDK4, K6. These proteins are P15, 16, 18, and 19. Okay? Got it? We're gonna have 2 families. The first family is of CDK inhibitor S, which having 3 proteins, 21, 27, and 57, and for an inhibitor for CD4 and CD6 are P15, 16, 18, and 19. Now, for example, if there is defect in the enzyme, like if there is defect in the inhibitor of the cell cycle, so what will happen? अगर inhibitor में मसला है तो cycle होगी नहीं. लाइक अगर इन सॉरी अगर इनहिबिटर्स में मसला है ना, तो क्या होगा कि हमारे पास डिफेक्टिव साइकिल होगी, डिफेक्टिव साइकिल की वजह से हमारे पास डिफेक्टेड सेल्स बनेंगे, और डिफेक्टेड हमारे पास सेल जो होगा वो बेकार हो जाएगा.